Hello, my sunshines. Welcome back to Chapter 22 on evolution. So in our human biology course, we've covered the body systems, their anatomy, the structures, and their physiology, the functions. And then we started going through general bio topics. So we hit mitosis, genetics. Now we are into evolution. And when you look at our course competencies, most of this is on the evidence of evolution. So we'll spend a little bit of time of general evolution info, scale of change, and then we'll spend a fair amount of time on the evidence of evolution. Okay. So let's get started. So the Earth about 4.5 billion years old. Very different than how it was today. We didn't have the same oxygen levels. They've gone up a lot since we had photosynthesis start. And then they were really high before we had decomposers. And now we're back where we are here. And evidence suggests that life has been here for about 3.8 billion. One thing to think about the whole time we go through this chapter is that 4.5 billion years, 3.8 for life, the changes that we're talking about occur, a fast change occurs in like 10,000 years, which is still an amount of time that we can't really understand. So we're talking about billions of years, which we really don't understand, like, in general, right? So when we say these things happen, they don't happen quickly at all. It's not just like, bam, a quick change is 10,000 years. Okay. So one thing we'll talk about a little bit is chemical evolution. So this is how different chemicals, molecules, have increased in concentration to where we're at now with those large macromolecules, the organic molecules of life that we've looked at. Okay. So small inorganic molecules join together to form larger, and then we get organic molecules that are like DNA, RNA. We get cells, true prokaryotic cells like bacteria, and then those cells get more complex. The DNA is enclosed in a nucleus. You've got membrane-bound organelles. So you've got a eukaryotic cell, and then multicellular organisms, right? There are still prokaryotic cells out there, obviously, bacteria and such. Our cells are eukaryotic, plant cells. Don't get too excited. Okay, so how could we possibly get chemicals, you might ask? Well, there was an experiment that was done by Miller and Urey where they put the molecules that were present in the Earth's atmosphere back 4 billion years ago, and they put in electrodes and boiled water, and what they found that formed spontaneously were small organic molecules. So that's pretty cool. Okay, so next let's look at scale of evolutionary change. That big picture evolution is macroevolution, where you have large changes over long time periods. That's what I was talking about a few slides ago. Microevolution, on the other hand, happens within a population very quickly over time. You can see this in a bacterial petri dish in ours, where you'll see changes at the genetic level, different frequencies of alleles. So evolution is a change in allele frequency over time. You can see that quickly in microevolution. Macro, it's these bigger scale changes. So this picture shows how you can have changes in alleles that give different characteristics of different Canis species. And then macroevolution, different species. One thing that we'll look at a little bit later, but this is not a picture showing trilobites becoming fish, becoming reptiles, becoming mammals, becoming humanoid. It's showing that they have distant related ancestors. Okay. Cool. Okay, basic terms in evolution. Big one is population. So a population is a group of individuals of the same species that are interbreeding. So group of individuals of the same species interbreeding. Okay. And the gene pool, that's the available alleles. So what are the different forms of genes that we have here? Available alleles. So different populations might have different gene frequencies, might have different alleles. Right? And so the picture below shows a group of flowers of the same species that are cross-pollinating. So they would be considered a population. Okay. So this is a really important general biology topic. Different ways that we have those microevolutions, the changes in allele frequencies in a population. First, genetic drift, that's just random. Random chance that some individuals bred and some didn't. Random. So for an example, here we've got the original population. 3 out of 7 are green alleles, 4 out of 7 are brown, and then somebody just squishes the green ones. So that population over time changed to be 100% brown beetles. It wasn't because the brown outcompeted the green. Green just had bad luck. We'll look at a couple of examples of that. No, we won't. So let's talk about them now. Bottleneck is one where you'll take out a lot of individuals, as well as a founder population where individuals from a larger population will move on to another area and interbreed, and the alleles that they bring might not be representative of the whole population, right? That has to do with gene flow as well. Individuals from one population moving to another, right? Immigration, emigration. Immigration / emigration. Okay, so this picture shows gene flow. Mutation. Mutation is the most important one here because that's where all genetic variability comes from. If there weren't different options of alleles, there wouldn't be selective pressures on those. And then natural selection is a big one. So here we'll look at this a little bit more with Darwin, but basically some genes are better suited for the environment. Those individuals are going to be healthier. They're going to pass on more of their genetics. And so their genes are going to spread in the population. So in this case, the selection pressure is that the chameleons can see the green beetles, so they eat the green beetles, and over time those brown ones blend in better. They become more popular. Right? That selection pressure was the predation by chameleons. You can also have sexual selection, which is pretty cool and can also be pretty bad. The Irish elk are an example of an extinct species where they kept picking larger and larger antlered males until they couldn't survive with their antlers. They were too large and heavy. So that was a runaway selection. Otherwise, you'll see different indicators of healthiness that females will use to select males. Usually it's the females picking because, as we looked at in meiosis, they put in more energy into their gametes, into the raising of offspring. Not always true, but most of the time. Okay. Cool. So what makes a species a species? There is not one perfect answer. Probably the most commonly used is the biological species concept of, do these individuals breed together? Does it happen in the wild, in their ecosystem? Right? Individuals that can interbreed. And produce healthy offspring that can also breed. So you'll talk about like prezygotic barriers and postzygotic barriers. Are you breeding at the same time as the other individual? In the same area? Do you have a certain call, like behaviorally? Are you going to look for a specific thing that if the other individual doesn't do, you won't breed with? The same season, the same time of day. And then postzygotic, is that offspring able to develop, and then is that offspring fertile? So common examples there would be like a mule that is not able to pass on their genes. Cool. And so speciation is seeing new species occur. OCCUI. So basically over time, if you have groups breeding by themselves long enough, they won't be able to breed together anymore. They'll be different species, right? Can a tiger and a lion breed together and form a liger? Right, yes, but they don't do that in nature. They don't occur at the same spots. They wouldn't use the same behaviors. There's a couple different ways this can happen. Allopatric or sympatric speciation are in the same place. Sometimes, like Grand Canyon, you'll have different types of ground squirrels on each side that can no longer interbreed because they can't cross the canyon. Fun. Very important. Mutations, let's look a little bit more at this. That's a change in the DNA sequence of genes. One thing to note here, usually mutations are going to be bad. An abnormal protein is generally not advantageous. Every once in a while, though, it is. But only if that mutation occurs in the gametes where we can pass them on will you see that to be heritable, right? So here you can see a different nucleic acid is going to cause either an abnormal protein or a non-functional protein. It won't make anything. We have a balance of checks in place to prevent mutations, but they still occur sometimes. Okay. So we could talk about this slide forever. I'm going to try to not take too long on it. We've talked a little bit about what natural selection is. It's a mechanism for evolution. And the first person to publish on this was Charles Darwin. Actually, there was another man, Alfred Russel Wallace, doing the same work, but Darwin is generally credited with all of the credit. But Charles Darwin sailed on the HMS Beagle, a boat from 1832 to 1836, and during that time he made observations about different biodiversity, different life. He went to the Galapagos, saw tortoises, mussels. A quite famous one is finches. And he looked at the shape of the finches' beaks and how different beak shapes had different diets. And so he hypothesized that that was due to selection pressures. Those beaks were optimized for the size seeds available. There's a pretty cool book called Beak of the Finch where a couple go and they measure beaks of finches over time and see how, as the rains change and different seeds are available, you'll see the beak sizes change. That change is evolution. Pretty cool. Okay, so now let's look a little bit more at the three ideas of how natural selection occurs. So first, you have genetic variability through different alleles, and that can get passed on. It's heritable. Second, more offspring are produced than will survive. And then third, the traits that allow those individuals to be more likely to survive will be passed on and become more popular in the population. So it makes a lot of sense. You can pass on genes, lots of offspring are produced, not all of them will breed. The more successful will breed more, you'll have more of their genetics in the future generations.